Hello everyone and welcome to our module on special growth requirements. In this module we're going to talk about bacteria that have specific environments where they prefer to grow. So first we'll talk about the obligate anaerobes. These are bacteria that only grow in environments that lack oxygen. Then we'll talk about the obligate aerobes. These are the opposite. These bacteria only grow in environments where oxygen is present. Next, there are the facultative anaerobes. These are bacteria that can grow in environments either with or without oxygen. And then finally, we'll talk about bacteria that only grow intracellularly. First, let's talk about the two different biochemical methods used to generate ATP for energy inside cells. The first thing we'll talk about is respiration, and this is what happens inside the cells in the human body. As you know, those cells use the electron transport chain to generate lots of ATP, but this process is aerobic, meaning that it requires oxygen in order to work. The other way to make ATP is called fermentation. This is where sugars are turned into acids. It does make ATP, but it makes less than respiration. But importantly, it's anaerobic. It does not use oxygen. And many bacteria and yeast use fermentation in order to generate ATP. The human body generally does not. Although when your muscle cells are deprived of oxygen, they can do a form of fermentation. This is how you get lactic acidosis. Aerobic organisms have two important enzymes that allow them to survive in environments that contain lots of oxygen, and those two enzymes are called superoxide dismutase and catalase. So bacterial metabolism in an oxygen environment produces superoxide radicals. These are written as O2 with a negative sign on them, and those are toxic to cells. They can kill the bacteria. But the presence of these two enzymes allows them to survive and get rid of that superoxide radical before it causes harm. Superoxide dismutase is going to catalyze the superoxide radicals into oxygen or hydrogen peroxide, and then catalase will convert the hydrogen peroxide into oxygen and water, which are obviously not toxic. So the bottom line is you need these two enzymes in order to survive in environments where you're metabolizing oxygen. Bacteria that are obligate aerobes must have oxygen in order to generate ATP. The only way they can generate ATP is through using an oxygen-based system. These bacteria are sort of like our own cells in the human body, which also require oxygen to generate sufficient ATP. For obligate aerobic bacteria, oxygen is the final electron acceptor during respiration, just like in the cells of our own body. The benefit of being an obligate aerobe is that these bacteria can generate lots of energy, more so than anaerobes. Also, these obligate aerobes must have superoxide dismutase, like I talked about in the last slide, because this protects these bacteria from oxidative damage. And some of the bacteria that are famous for being obligate aerobes include Pseudomonas, Mycobacterium tuberculosis. Tuberculosis loves the lungs because it loves oxygen, and Nocardia, which is a cause of opportunistic infections. Obligate anaerobes, on the other hand, lack either catalase or superoxide dismutase. And it's very high yield to know that obligate anaerobes are common among the normal flora of the gut and mouth. About 99% of the fecal flora are anaerobes, and there are 100 times more anaerobes than aerobes in your mouth. This is sometimes a point of confusion for students because you may say, wait a second, there's a lot of oxygen present in the mouth. Why are these bacteria able to live there if they're obligate anaerobes? But you have to remember that the mucosa of the mouth and the GI tract is a very complicated area, and there are pockets where there are no oxygen, and that's where these bacteria can live. In general, they don't cause communicable diseases because it's hard to transmit them from one person to another. As I said before, they often live near the mucosal surfaces, and the main time when these bugs cause infection is when surfaces break down. So you often find anaerobes in abscesses. You can imagine that if you've got anaerobes living on the surface of your mouth and you develop a dental abscess, that those anaerobes can get inside and there's very little oxygen inside those abscesses, so they can grow and thrive and cause symptoms. Another high yield thing to know about anaerobes is that aminoglycosides don't work. They're ineffective. And that's because aminoglycoside antibiotics require oxygen to be transported into cells. Some people just remember the O in aminoglycosides for O2 to remind them that oxygen is necessary for these antibiotics to work. Obligate anaerobes use fermentation to make their ATP. They don't do respiration. That's because they don't use oxygen. And they have a couple important byproducts of fermentation you should know about. First of all, they produce gases like carbon dioxide and hydrogen. They also produce short-chain fatty acids from fermentation. These are molecules with names like acetic acid and isobutyric acid. And the result of these gases and short-chain fatty acids is that you get a foul smell. And if you've ever seen anyone who has a pustule and then pops it, you know that it often has a foul smell. And that's because these anaerobic bacteria are using fermentation and creating these byproducts, which are what smell bad. So I've written on this slide the names of some of the anaerobes that you should definitely know. The first one is actinomyces. This is found in the gums and it's often a cause of dental abscesses. The second one is Bacteroides. This is found in the GI tract and it often causes abdominal abscesses. So patients who have appendicitis may have an abscess in their appendix and this bug is often found. And then finally, all the Clostridiums are anaerobic. So botulinum, perfringens, tetany, these are all anaerobic bacteria. There are a couple of important infections where anaerobic bacteria play a key role, and let's talk about those now. So abdominal abscesses or perforations often involve anaerobic bacteria. When you have a breakdown in the mucosal layer of your GI tract, you will get an abscess that contains many gram-negative GI flora like E. coli, but it will also contain B. fragilis, which is an anaerobe, and B. fragilis loves to live in those little abscess pockets with no oxygen. B. fragilis is resistant to many antibiotics, but it is covered by metronidazole or flagyl, so you will often treat patients who have diverticulitis or appendicitis, which are abscesses in their abdomen, with metronidazole plus some agent to cover gram-negatives. Usually that's ciprofloxacin or quinolone. Another infection where anaerobic bacteria play a key role is aspiration pneumonia. So when you aspirate vomit, mouth anaerobes will enter the lungs. These are bacteria like Peptostreptococcus and Fusobacterium and Prevotella. And this is why the antibiotic agent of choice for aspiration pneumonia is clindamycin. Clindamycin is an antibiotic that covers anaerobic bacteria. We say that you use clindamycin for anaerobic infections above the diaphragm, and the lungs count as above the diaphragm. And you use metronidazole for anaerobic infections below the diaphragm, like abscesses or perforation. And these are probably the two most important clinical scenarios where anaerobic bacteria play a role, and you should definitely know about these two situations for step one. Facultative anaerobes are bacteria that can live without oxygen, but they use it if it's available. So these bacteria are capable of performing respiration and fermentation to generate ATP. There's something called the Pasteur effect, which is the generation in the microlab that when you give oxygen to these bacteria, you inhibit fermentation and they begin performing respiration to generate energy. And many common bacteria fall into this category of facultative anaerobe. This includes staph and strep and E. coli and a lot of common bacteria. There's another class of aerobic, anaerobic bacteria that's not really that important in medicine, but you may have heard about this in a biology class in college. These are called aerotolerant anaerobes. These are similar to facultative anaerobes, but they always use fermentation, even in the presence of oxygen. Bacteria like this are rare, and there really aren't many examples relevant to clinical disease. I just mentioned this because you may have heard about this in a biology class. Final topic in this module are obligate intracellular bacteria, and these are bacteria that cannot synthesize their own ATP, so they have to live inside a host cell and steal energy from that cell. Most of these bacteria do not gram stain well because they're always living inside other cells, so it's hard to get them on a slide and get a gram stain of them. They're also difficult to grow because you need to use a cell culture. You can't just put them on a plate of nutrients and watch them grow. They need other cells to infect. And the two most important obligate intracellular bacteria for you to know about are Rickettsia and Chlamydia. So Rickettsia causes Rocky Mountain spotted fever. We'll talk about this in another module. But the one thing you should know is it's diagnosed either by clinical symptoms or with serology, which are antibody tests. It's not diagnosed by culture, and that's because it's intracellular and it's hard to grow. Second obligate intracellular bacteria is chlamydia, which causes sexually transmitted disease and some other infections. And this is also not usually diagnosed by culture because it's very hard to grow. You diagnose chlamydia infections usually with DNA testing called nucleic acid amplification testing. Finally, I'll mention that there are a lot of bacteria which are called facultative intracellular, which means they can live inside other cells if they need to. And this is often very important to the pathophysiology of the bug. So for example, Mycobacterium, like Mycobacteria tuberculosis, likes to live inside macrophages. And a very important part of the way tuberculosis causes disease is that it infects macrophages, grows, and then ruptures the cell. And there are many other bacteria which commonly live inside of other cells. This includes Legionella, Salmonella, Neisseria, Listeria, Brucella and Francinella and Yersinia, which are all zoonotic infections, meaning they come from animals and infect humans. And you'll notice that many of these bacteria live inside of macrophages, and that's because the macrophages try to phagocytose them and kill them, but the bacteria have developed ways to evade that, and therefore they can live inside the macrophages for a period of time. And that concludes our module on special growth requirements.